Welcome to NISS-CANSSI Collaborative Data Science webinar series. So my name is Saman Muthukumarana from Department of Statistics and University of Manitoba. I'm a professor in statistics. So this is the fourth webinar organized by NISS-CANSSI Collaborative Data Science webinar series. And I would like to start by thanking the National Institute for Statistical Sciences and the Canadian Statistical Sciences Institute for sponsorship of this online webinar series. So this is a great initiative where statisticians and data scientists and applied statisticians and scientists come together and talk about how statistics and data science evolved through collaboration. So goals of this series is to showcase their experts from various fields who tackle complex data challenges through interdisciplinary teamwork by featuring collaborators from data science and areas of science in broad perspectives. So you will hear some interesting stories today as well. And if you have any ideas about how we can enhance this webinar series, and if you have ideas for speakers, even maybe yourself, please feel free to reach out to us. And so with that, I would like to introduce our speakers today. We have two prominent speakers who will really talk about their collaborations. Dr. Laura Cowen, a professor of mathematics and statistics, and also she is the acting dean of science at University of Victoria, BC in Canada. She's a well-prominent statistician as well as very ecological scientist as well, who has a lot of extensive collaborations with industry and government partners. And with her, we have Dr. Patrick O'Hara. He's a senior scientist at Integrated Marine Spatial Ecology Lab, as well as Institute of Ocean Sciences. So these two pair will talk together how they have working together in this collaborative projects. So title of the talk today, you are in the right place at the right time today. And they will talk about how statistics and data science with ecological applications shape up their collaborative work. And if you have any questions throughout the talk, please use in the Q&A option. If you go to the Zoom bottom, you will see Q&A, and you can use that Q&A option, and you can put the question there. And then during the question period, I will read out your question, or I will call your name and unmute and ask the question if you ask that request. With that, I will pass over to Dr. Laura Cowen and Dr. Patrick O'Hara to take their session started.
All right. Well, thanks very much for that intro, Saman. It's nice to meet you virtually, at least. Yeah, so Laura and I are here to talk about some of our collaborative work that we've been doing on ancient murrelet and their conservation. Here's our study species and a chick. So a little bit about ourselves. I'm a conservation scientist. And as a conservation scientist, one of my main goals in trying to achieve some sort of conservation goal is to identify and define knowledge gaps and assign some sort of priority to those knowledge gaps. And this requires many skills. So I have a lot of skills, but I'm just generally skilled. I'm not expert in anything, so sort of a jack of all trade, but I do recognize the need to bring in experts for various parts of whatever it is that we're trying to conserve, whatever project conservation. And so Laura and I have, Laura has a background in ecology as well, and she brings in some skills that are very complementary to what we're trying to do in the conservation world. Laura.
Hi, everybody. I don't know what's going on with my camera, so apologies for this. But yeah, I've got a background in biology. I've done seabird field work, so I can understand Patrick's world to some degree. And then I work on ecological problems as a statistician, developing methods, but also applying complex models for ecology. And we'll be talking about 2 different projects that Patrick and I have collaborated on. So back over to you, Patrick.
Great. So yeah, I'm going to provide a bit of an ecological framework or conservation framework, and then Laura will talk about the nitty gritty and some of the models that we've been developing to achieve some of these goals that I'll describe. But first, I'm going to introduce the taxa that we're working on. Seabirds in general have been declining. Populations have been declining worldwide, and in some cases by as much as 70%. As outlined in this paper by Michelle Polechny. And in this paper, they make the case that fishing, competition with fisheries, is one of the biggest reasons for these declines. And another big aspect of fishing is so-called incidental take, where birds get caught up in fishing gear, such as gill nets, or they go after the lures. A lot of albatross and shearwaters go after fishing bait and get caught by long lines. Another big threat to seabirds worldwide, of course, is climate change and all the factors that go with climate change. Habitat loss has been a well-recognized over the years as a big threat, particularly to breeding habitats, where most of the work is concentrated. Introduced species is another largely breeding threat because most seabirds mostly breed on the ground in isolated areas, such as rocky islets or in cliff faces, but are not immune to introduced species such as rats and raccoons. And that's a big deal, particularly along the BC coast. Pollution, of course, we've known for a long time. Pollution is a big problem for seabirds, in particular, oily substances from vessels, oil spills, chronic oil spills. And plastic is an evolving threat. And then noise is also an evolving threat in terms of our understanding and light pollution from vessels in particular, and offshore oil and gas. Okay, so we're going to talk about, sorry, to move this over a bit. Okay. Okay, so we're going to talk about, sorry, I went back, going forward. Okay, precision. We're going to be talking about precision mostly in our talk today. So estimating precision is fundamental to conservation science for identifying and characterizing key areas spatially and for detecting changes over time. In particular, Laura and I will discuss how we distinguish locations in movement data to identify important foraging areas, so areas that birds concentrate at, so that conservation scientists such as myself can identify and mitigate threats to these areas. Seabirds can be particularly vulnerable to threats while foraging, as they tend to concentrate in areas, making them vulnerable to higher frequency, though often smaller spatial scale threats. In the second part of our talk, we'll discuss how precision improves our capability to detect changes in populations over time. This helps us clearly identify populations at risk, particularly in a BACI or BACI, before-after control impact design frameworks. We can then identify impacts associated with particular threats or measure the effectiveness of conservation measures we might have implemented towards their conservation. So movement data are typically geopositions and time. And so movement characteristics can be measures such as distances between points or time between points, both, or turning angles. And with these movement characteristics, we can infer behaviors that underlie or cause these movement characteristics. Now, early on, we use clustering techniques looking for area-restricted search patterns where birds change their movement characteristics in response to being in an area where they think there's elevated or enhanced productivity. And this has indicated to us that they might be foraging in these areas. But today we'll be talking about more sophisticated approaches to identifying foraging areas or other behaviors of interest based on movement data. Of note, there are two important annual cycle events that make this particularly important to us as conservation scientists. And one of the events or concepts is that we have central place foraging. And central place foraging happens particularly during the breeding season, where birds have to return to their colony either to provision chicks or incubate or lay eggs. This constrains foraging birds to within a certain distance from the breeding colony, and that means that there is a trade-off in terms of energy and time spent foraging and breeding success. The non-breeding, and the other concept is that we have non-breeding stopover or overwintering sites. And this is where birds migrate often large distances to locations where they expect to find sufficient food to move on or survive their winter. And a really good example of this here off of BC is the sooty shearwater. They breed in the southern hemisphere in islands around New Zealand. They're called the mutton bird, because they're very important to the Maori and others who harvest them. And then during their austral winter, they come all the way up to off the coast of British Columbia and exploit enhanced productivity that we have up here during our summer. And this happens to an extent that they become the most numerous seabird off the west coast of Canada during our summer. So here's an example where we define foraging locations based on movement data for kittiwakes on Middleton Island here, right here. Oh, it's actually nicely labeled, Middleton Island in the Gulf of Alaska. Here we're able to show how foraging trips increased in size in association with the marine heat wave in 2014 to 2016-ish. That's the so-called blob. And this meant that central place foraging birds had to fly farther. You can see as you move into 2016, 2017, and 2018 during the heat wave to cover energetic costs of breeding. And so we moved, this is locations, and once we were able to identify foraging areas, we were able to show how far they had to fly in order to sustain themselves and produce chicks. And another really good example, I think, is that of using movement data in this case. Oh, why does it do that? Is another great example of using movement data to identify foraging areas. In this case, movement data from over 21 species and 105 colonies, breeding colonies, we used to identify an important foraging area in the middle of the North Atlantic outside the jurisdiction of any country. So you can see all these locations globally where they bred, and then during their winter, they moved into this small area in the North Atlantic to supposedly because of expected enhanced productivity there and foraging opportunities. This was part of an initiative labeled Biodiversity Beyond National Jurisdictions, or BBNJ, under the OSPAR Commission. In the upper globe, figure shows how birds from colonies across the North Atlantic moved into the Nasties, or the, what's it called? The North Atlantic Current and Evelyn Sea Basin, MPA, or Marine Protected Area. And so with these data, they were able to identify this area. And the next thing they're doing right now is that they're trying to characterize that area using external variables. And so the question is, what makes this area so attractive to birds now? And the assumption, of course, is that there is, as I mentioned, an expected enhanced productivity in this area. So the question now is why? And characterization usually involves using explanatory variables that can both be fixed, such as depth or distance from shore, or ephemeral, such as sea surface temperature or chlorophyll biomass. And I'm showing you here an interesting example of ephemeral data. This is sea surface temperature that is used to indicate the location of fronts, and in particular convergent fronts. So you have water masses coming together, and that retains nutrients and productivity and becomes an area of enhanced productivity. And this particular front is associated with the North Atlantic Current, which is an extension of the Gulf Stream. So you can see that the fronts, although ephemeral, are still predictable over time. So we have the same sort of fronts that are predicted in shown in 1983 are fairly consistent with what happening in 2019. The concern here, of course, is that it's a thermohaline current, generated current, and it's susceptible to changes due to climate change. So this could fail at some point. Precision is also important for us in terms of measuring changes in population over time, particularly by reducing the confidence interval of population estimates and trends. One important way of doing this started with Leslie matrices way back when, and I'm not even sure how long ago this started up, where variability was parsed out into age-specific rates, which allowed us to explain and simulate variability in population estimates, i.e. lambda, in a time-specific fashion. And then since then, we've moved into other more sophisticated modeling techniques that involve matrices and transition matrices, such as capture modeling, teasing apart recapture reciting rate into survivorships and mortality rates and recapture and reciting probabilities. And then we also have state space models that separate processes into observation process, i.e. what's being observed, and the state process, which is the underlying biology that drives what we're seeing. And what we'll be talking about today will be a multi-event modeling where we use state transitions that are observed with some degree of error, trying to estimate that error. And here's an example using a matrix, transition matrix. And this is done by Peary et al. In 2006, and it's based on data collected on the marble murrelet, which you can see at the top, which is another protected bird, threatened bird. And in this case, they combine demographic population time. In this case, they had 4-stage juvenile, 2 adult substages, and breeding adult stages. They used mark recapture techniques to estimate survivorship and recruitment, and integrated these results with population estimates based on at-sea data. In the top right figure, you can see the estimated birth rates in black as a proportion of breeding adults and the estimated total populations in the region. They were able to tease apart local birth rates and recruitment or immigration from outside populations in their results. In the lower figure on the right-hand side, you can see what their estimated lambda based on their matrix model on the left, and that is lower than 1, while lambda based on their at-sea populations indicate that local reproduction rates do not support the lambda based on at-sea models, concluding that their population acts as a sink to marble murrelet populations outside their region. This, of course, has implications for the conservation of their local murrelets, but in addition, for murrelet populations outside their study region that might be losing breeding adults to the sink population in California. Okay, so that's my intro, and now Laura's going to take it away, and I'll be doing the slides.
All right, so we just want to introduce you to the ancient murrelet. It's quite an interesting bird.
Oh, sorry. Yeah,
50% of the population actually is hosted in Canada on the Haida Gwaii Islands. And so this is why it is a species of interest, because it's so concentrated in one area. Ancient murrelets are quite vulnerable to predators, rats and raccoons, mainly because when they come on shore to breed and to lay their eggs, that is where they are vulnerable. So most of the time they spend their life at sea, but in order to do their breeding, they actually burrow into burrows, lay their eggs, and this is what exposes them. Anything more to say there, Patrick? I kind of jumped.
No, sorry about that. I'm confused by the layout here. That was my slide, but you covered it great.
Okay, good. All right, next slide.
Okay.
Okay, so the first study that we want to talk to you about is to do with movement modeling. And this was really led by a master's student in geography, Vivian Pattison, that Patrick and I co-supervised with Chris Bone. Vivian worked very closely with Canadian Wildlife Service, and what we were trying to determine is, can we use movement data to look at ancient murrelet behavior on foraging trips? And if we can do that, can we characterize the behavior and determine what exactly they were doing on those foraging trips? And this paper was published in IBIS. Next slide. So this gives you an idea about how we can access ancient murrelets. It's dirty work. We have to, what's called grubbing, get our hands dirty, go in, pull the birds out of the burrows, and then they attach a GPS tag. And the lower left-hand corner, you can see the GPS tag attached to the back of the murrelet. The tags store the data, and so in order to retrieve the data, we have to retrieve the bird and the tag and download it. So it's tricky work to do. We also can take blood samples in order to determine sex. And there is a bit of a balance between the data and battery life. So we played around with this. In 2018, we took a location every 30 minutes, and in 2019, we tried every 20 minutes. The other thing that the tag records is time of day, and this helps us determine what behavior is happening, and I'll talk about that a little bit later. And in 2019, the tags were also able to get immersion data, so whether the tag was wet or dry, and we were able to sample that every 5 seconds. And we defined a trip as the murrelets being away from the island for more than 24 hours. So this gives you an idea of the different pathways for the individuals. In 2018, they went to Ramsay Island, and in 2019, they were out in George Island. Both of these islands are on the east coast of Haida Gwaii, and all of these birds are using Hecate State quite widely. Next slide. So in order to analyze movement data, typically people use hidden Markov models, and I'll give a very, very brief introduction to hidden Markov models. The idea here is that the model is broken up into an observation process and a state process. The state process is underlying, it's latent, and this is our unknown murrelet behavior. And then the observation process is the time series of locations, and this is informed by the state process. And for us, these series of locations, we can determine the step length, which is just the distance between locations, as well as the turning angle, which is just the change in direction of travel. Next slide. So the models, we use different models for each year. We tried to keep the first stab at this fairly simple, and so each year also correlated with the quality. Step length typically in the literature is modeled using a gamma distribution, and turning angle is typically modeled using a von Mises. And Vivian implemented these models using this R package Momentu HMM. Next slide. So in order to determine the number of states in the model, which to begin with is unknown, we followed this iterative process and we fit models with 2, 3, or 4 states. And we also included covariates or did not include covariates. So we looked at a whole bunch of different models. And then we used AIC and pseudo residual plots in order to assess the fit and finally select a model. And eventually, once we had our chosen model, we interpreted the states using the ancient Murrelet biology. And so this is really where we take models and combine them with the domain knowledge that Patrick has. Next slide. So we ended up choosing a 3-state model, both for the fit and the parsimony. And we did include time of day and the dry index as covariates to inform the states. So the 3 models, the 3 states, the first state was very long step lengths, and this corresponds to fast movement, variable turning angle, and we notice that this is always when the Murrelets were leaving or returning to the colony. So we called this state the transit state. The second state had shorter step lengths, so slow movement. It was concentrated in one location, variability in both the step length and the turning angle, and so we called this state the foraging state. And then the third state had shorter step length, so slow movement, and also persisted in one direction, so we called this the resting state. Next slide. So the interpretation of the behavior states is really dependent on biological knowledge and then the temporal resolution. If we can identify the foraging states, then we know where the foraging areas are, and this can inform conservation policies and planning. The immersion data and the time of day were used in the model, but they also were used to validate the model. So for example, if we know the individual is wet or the tag is wet, then likely the bird is diving and they dive when they're foraging. And similarly, if we know that the time of day is during the night, we know that ancient Murrelets typically are not foraging. And so this tells us that this is the resting state. So we have more future work to do on this. We think there might actually be a 4th state, which is where the birds are out on socializing. They tend to just form in rafts just off the islands, and they do a lot of different movements. So it's hard to tell without additional data. And then the other thing we'd like to do is use environmental variables such as sea surface temperature to try to predict where the important habitats exist, again, for conservation planning. And then the next study I wanted to talk about is a more recent study that we've done on population trends. This was led by Shay Ahuenajed, who was one of my master's students. Anecdotally, on the island, biologists and field workers have noticed that there seems to be a decline in the population, and specifically the non-breeders. So non-breeders typically will come on the island, and they're like teenagers. They like to socialize, they like to make a lot of noise. And in the more recent years, the island has gotten quieter and quieter. So we suspect that there are fewer and fewer non-breeders. So to take a first look at this, we wanted to determine if there was a survival difference between breeders and non-breeders. And I should mention that this work is in collaboration with Simon Bonner at Western University. So next slide. So just to orient you, this is a map of the Queen Charlotte Islands, or Haida Gwaii, and the island for this study is East Limestone Island that you can see there in the red circle or the orange circle. Next slide. So the data here is collected by the Lesquike Bay Conservation Society. They became interested in monitoring ancient murrelets in 1989. They played around a lot with their methods of monitoring, and so we are only using the data from 1996 to 2002 when the methods stayed consistent. And up in the top, you can see what's called a mist net. For ancient murrelets, we have to use a bit of a more burly net, which I think they call a goose net, but it's the same idea. A bird will fly into a net, drop down onto the ground, and then we can actually handle the bird, tag it, and measure it. The sampling occurs each May when the birds are on the islands breeding and socializing. And in table 1, you can get a little bit of idea of the numbers of birds we're working with. These are the new banded ancient murrelets between 1996 and 2002. Next slide. So the reason we are, or one of the issues with this data is that there are observation errors. So not all birds were assessed for their breeding state. And even if they are assessed, the breeding state comes with errors and when we try to assess it. So there are, oops, back one, sorry. I'm not sure what happened there, Patrick. Keep going. There we go, there we go. So we determined 4 different types of observations. A 0 is if it was not observed or not captured. A 1 was that it was assessed to be a non-breeder. A 2 was that it was assessed or ascertained to be a breeder. And then a 3 was that we did capture the bird, but it wasn't obsessed. And so in typical capture-recapture studies, the data becomes a time series of 1s and 0s for whether the individual was captured or not. But in this situation, we get a time series of 0s to 3s depending on our observations. Next slide. So how exactly do we ascertain the breeding state? Well, sometimes birds are found in their burrows with an egg or a chick present, and so then we are pretty certain that those birds have been breeding. But the other way to do it is if we capture the bird using those mist nets, we can check for what's called a brood patch. And a brood patch is just an area on the underside of the bird. It's highly vascularized, which means it brings a lot of heat to the area, and this helps heat up the egg during incubation. And what previous biologists have found is if the brood patch is more than 11 millimeters, it is likely a breeder. Next slide. So once again, we're going to be using hidden Markov models, and Pradel developed what he called the multi-event model, which is just a hidden Markov model when there is state uncertainty. And so indeed, trying to determine whether we have a breeder or not, we have state uncertainty. And our 2 states are the survival, so whether the bird is alive or dead, or the breeding state, so breeder or non-breeder. The other nice thing about these models is we are able to estimate the transition probability between states, so going from non-breeder to breeder. Next slide. And we haven't had any equations yet, so I thought we should throw one up. This is the likelihood for a hidden Markov model. It's very nice to use and very simple because it's really just a product of matrices. The delta here is just the initial distribution, and I'll describe that in a moment. The P matrices are for the observations or the captures, and the gamma matrices determine they try to model the survival and the breeding state transitions between times. Next slide. So for our case, the delta is just the initial probability. which here is just the initial probability of being non-breeder. We also have the initial probability of being a breeder, which is just 1 minus this pi. And then we also have the probability of being dead, which for all the birds that were captured was 0. Next slide. Then we've got the transition matrix, and it models the probability of transitioning from state I to state J. And here our states are either non-breeder, breeder, or dev. Next. So this is our transition matrix. And along the top, we have our 3 states, non-breeder, breeder, and dead. And along the side, we also have our states. So going from non-breeder to non-breeder, you can see that that component, you'd have a survival component and then a transition, but it's actually the probability of just staying as a non-breeder. If we're going from non-breeder to breeder state, then it survives as a non-breeder, but then transitions to being a breeder. And then if the bird is already in the breeding state, then we're just modeling survival or death. Next slide. The observation matrix is the probability of having our observations, so those 0, 1, 2, or 3, given the underlying state. And so our observations I've described before is that we were assessed as non-breeder, assessed as breeder, not assessed or not observed. And so to break this down a little, I drew out a tree matrix. How do we get these observations? Well, the individual is either captured or not captured. If it's captured, it's either classified while it being a breeder or not classified. Similarly, while it's a non-breeder, it's either classified or not. And then if it is classified, it's assessed to be a breeder, or it's incorrectly assessed to be a non-breeder. And similarly, if it's a non-breeder, it's assessed to be a non-breeder or incorrectly assessed to be a breeder. Next slide. So if we look at this observation matrix and break it down, we've got the possible observations I've put along the top row, and the underlying states are along the column. And so if we, next slide, I think I break this down, yes. If we look at going from breeding to and not observed, we just have this 1 minus p, which is our detection probability. The psi is our assessment probability, and the deltas are all of our classification probabilities. Next slide. And so the model, we just have that model for the state transitions, and this is just categorical depending on that gamma matrix. And the observations are also modeled as a categorical dependent on that P matrix. All of this was done using Nimble in an MC-MC framework, and our priors were all uniform 0, 1 because they're all probabilities. So here is some of the results. These are preliminary. Shay just finished her master's, but I know there's work to be done. And so these are all of our parameters, and then we've broken it into breeder and non-breeder. And what you're looking at is the parameter estimates and the credible intervals. And next slide. So I'll go through them one by one. The detection probability, it looks like there is lower probability for detecting a breeder. And I suspect this is due to our sampling mechanism, because non-breeders hang out on the island and socialize, whereas breeders are trying to get directly back to their burrows. I think there's just more opportunity of catching a non-breeder. Next slide. The deltas, or whether we are correctly classifying them, and the credible intervals overlap, so there's not a lot of difference in those classifications of breeders and non-breeders. Next slide. For assessment, whether they go through the assessment, the non-breeders again have a higher rate, and I suspect this is probably due to our sampling mechanism. and then next. We did look at survival. This is, you probably note that this is constant survival over the entire study, and we did not pick up a difference in survival rates. And then finally, along the bottom there, whoops, back one, sorry, Patrick, is just for interest, this is our transition probability going from either non-breeder to breeding state or non-breeder to non-breeder state. And these are just one minus each other. Given that non-breeders are typically in the non-breeder state for 2 years, this is what the biologists tell us, this to me makes sense that it's around a 50% probability of transitioning. Next slide. So conclusions and future work. Survival likely varies over the years. We had it constant in this model, and so we'd like to bring in possibly some environmental covariates to inform this model and have it variable over the years, especially if there is a population decline that we're trying to pick up. We also think there's likely annual variation in the assessment data. This really depends on who's on the island that year doing the assessment. And then we'd also like to extend the model to try and estimate population size and eventually to look at trends in population. Next slide. And I think this is back over to you, Patrick.
Yeah, okay, thanks. So in summary, for ANMU or ancient murrelet conservation, we have shown 2 examples where we increase precision, in particular spatial precision, by identifying locations where individuals are likely to forage. This allows us to reduce our focal spatial polygons and hone in on those important foraging areas, spatially on maps. And we can then identify some of the key threats that we think these foraging habitats are facing and try to mitigate against them. And we can also characterize these habitats with external variables to understand why these habitats are so important. And we can then integrate locations and habitat characteristics into a predictive modeling framework to help fill the information gaps, because we can only put out so many GPS tags at a time, as well as use validation techniques in these models. We can use validation techniques to estimate confidence in our understanding of why these habitats are so important. As well, we can use predictive models to help us detect changes in the locations of these habitats over time, particularly with concerns with climate change. For temporal population trends, we've used HMMs again. This is purely coincidental that we're using HMM for both of these examples, but just the way it works out. And we used HMMs in a multi-event model to parse out constituent rates driving variability in population estimates, or lambda. With this model, we estimate and identify important constituent rates and reduce error associated with misidentification of life history stages. And we can associate these rates, as Laura mentioned, with external factors and ultimately incorporate rates and factors and again in a predictive modeling framework for similar reasons as we did for our foraging habitat, or as we plan to do with our foraging habitat models. So this increased precision and increased confidence in our estimates in both cases allows us to better protect key habitats, detect changes that should either alert us of a need for increased conservation measures or allow us to measure conservation measures we have already put in place. And with that, that's the end.
Okay.
Oh, sorry, we got a thank you slide. We can't, well, I'm not sure. It's not showing up. Oh no. We have an acknowledgement slide and it's not showing up. Just, I don't know what happened to it.
Okay.
There's a whole bunch of people we should acknowledge, particularly the Council of Haida Nations, Parks Canada, Canadian Wildlife Service, Western University, UVic, Institute of Ocean Sciences. It's all supposed to be there on the slide. Apologies for that.
Okay, thank you so much, Patrick and Laura. It's a very interesting talk. It's great to see how the statistics have knowledge has combined with these challenging ecological problems. So with that, we will have a Q&A session now. So Scott Pearson is asking, for your transition matrix, do you ever have skipping as a state? I don't know if, for instance, ever skip breeding season. So maybe.
Yeah, this is a good question. For our model, we did not have this. So once they are a breeder, they're always a breeder, or they attempt to breed. But certainly in other species, you could have a skipping state. And in fact, Saman and I are working with a PhD student on Dolly Varden fish, where the state switches from silver to spawner back and forth. And this is a tricky situation because you don't always know what state the fish is in. So certainly you could change your transition matrix and incorporate a skipping or transitioning back and forth.
Yeah, thank you, Laura. That's great. Are there any other questions? So if you have any questions, please use chat option or Q&A. You can simply post your question under Q&A section. And maybe I will have a question until people grab some ideas for questions. So maybe I would like to hear a little bit from both of you, Laura and Patrick. You had this spatiotemporal modeling where data will come in over space and time both. And this is very, very common in ecology, but also challenging, especially data collection. And can you talk about, because there are a lot of technological ways to collect data, sometimes observational data, experimental data, and what are the common challenges you have faced in dealing with this data?
Patrick, do you want to take that one? The challenges of working with birds and tags and all of that?
Yeah.
Laws.
Yeah, I guess in the field, I mean, there's lots of, where do you start? The big challenge, is for us is you want to attach these devices to these birds in a way that doesn't affect the behavior of the birds. So you're putting something on these birds and you're hoping that they go about their day in a normal fashion so that you capture data that's representative of the larger group of birds. And generally we tend to minimize the size of the tag as much as possible, but of course then you run into other problems when you do that, because the biggest thing in these devices is the battery. And so you have to really decide what exactly it is you want to collect in terms of the data. And Laura touched on this. We want it to be the high enough resolution that we can infer these behaviors over the scale of a single foraging trip, whereas other devices you may want to look at other aspects of their ecology, and you would change the resolution of the data that's being collected. And the other big challenge with these GPS data tags is that you have to recover the device to get those data. And I guess, and one of the really interesting things I think working with Laura is really trying to get at the underlying states. So there's a lot of back and forth. We take a look at the model, and then we think about the biology of the bird and what we know about the bird, and then we go back to the modeling. And so that's, and one of the outcomes of that iterative thing, as Laura mentioned, is this fourth state that we think could be in the data. And I think the only way we're going to really resolve that is by collecting more data and doing more modeling, because as you could, we're really limited in our sample size early on, but since then we've got quite a few more years of data. And so we're hoping to be able to identify more states, and maybe, as Scott says, maybe we can start exploring the skipping thing as well.
Thank you, Patrick. Yeah, that's great. And we have more questions here. One from Hwang Ho Kim. How are you estimating parameters of the transition matrix?
Yeah, for the first model, we implemented it using the package in R, and that is classical frequentis package. So it's just using some form of a maximization to estimate the parameters. And in the second one, Shay implemented it in Nimble, which, and we were just using an MC sampler. So that's how we were estimating those parameters.
Okay, thank you, Laura. And then we have another question from Scott Pearson. Your lambda is quite low and would suggest a decline in population. Are there other data you can use to inform lambda? Are you considering using IPM?
Yeah.
Good question.
Yeah. I think the short answer is yes. That is a good approach and one that I think we have that kind of data that we could implement IPM for Lesquik. And it's not surprising that lambda is below 1 for this particular population. I think Tony Gaston predicted it back in the 90s that this colony was in trouble, in particular because of raccoons at the time. So it does kind of confirm what Tony was already suspecting was happening out there. And Laura mentioned about the decline in non-breeders, and that seems to be a real thing.
Okay, great. We have another question from Ghazali Farjan. Have you examined potential impacts of climate change on their population dynamics, such as shift in distribution, migration timing, or reproductive behavior? Also, have you observed any emerging infection diseases or novel pathogens in this species that may be linked to change in climate change?
That's a good question.
I don't know, has there been any work done on diseases with ancient
Well,
Murrelet?
The research area, Laura work on a different human diseases, but that's interesting, yeah.
Yeah,
Wow.
I mean, we're always aware of and on the lookout for, avian influenza. I'm not the person to ask necessarily, but I'm not aware of that being a problem yet on these colonies, but that's not, I mean, these things, when they happen in a big way. So, so far so good, but that doesn't mean they're safe from it. Linked to climate change, that's an interesting one. I think there probably is, but we're not there yet in terms of associating any of these.
Okay. Are there any other questions? Oh, there is another one, Adam Wisme. Which states in the models are most sensitive? In other words, how much of future dynamics are affected by changes to a given state?
That's a good question.
Good question. I don't think we've looked at this, but do you
No.
have a sense, Patrick?
Well, I mean, typically with seabirds, they're most sensitive changes in adult survivorship because they tend to be long-lived birds, and they put disproportionately, when you compare them to other bird taxa, they tend to put a little bit less stock in each reproductive event. And the idea is that they're supposed to have as many reproductive events as possible. So that's classically what we're looking for is they're typically more sensitive in terms of tweaking adult survivorship, but we haven't done that yet. That's something that we could do over the next little while now that we've developed these models and do some simulations and see whether or not. But keeping in mind that right now we just have this Leslie matrix, we have this multi-event model for one specific colony. It's not for the entire breeding population in BC. So, but it still would be interesting to look, I think, to simulate this particular population and see, how they're most sensitive to changes in the different constituent rates.
Mm-hmm.
Any other questions? We have 4 minutes left. Maybe I will have one more question while both are here. On the Laura, your state space modeling framework, you had this 3-state model. I know it's not an easy model fitting process. And how did you deal with this? How many states to be accounted? And
For the movement model?
Yeah.
Yeah, so Vivian did a lot of exploration, and I think I mentioned we followed this iterative process that was suggested in the literature. But this is really tricky, how to decide how many states to put in the model. And so we had this balance of fit versus parsimony. Could we even explain the states? So
I think this is where perhaps you use domain knowledge coming
exactly. Yeah. Could we explain a fourth state? Maybe, but we felt like we could certainly explain the 3 states, and the 3 state was fitting fairly well. So you're right. It's a balance of the statistics, but you really need that domain knowledge.
Exactly. I think that's very important in collaboration, and that's exactly what this data science where collaborative work is very important. And with that, are there any other final chance for any questions for anyone? Seems like, oh, there is another one. Monique Lance, is anyone studying diet simultaneously? It appears there are definite important foraging areas based on tags. Was there individual variation?
I think there's been a lot of work on diet, but I'll let Patrick, you go ahead.
So I missed the last part of the question.
Okay, so it appears there are definite important foraging areas based on tags.
Yeah.
Was there individual variation?
Right. Yeah, there definitely. There's individual variation. Okay, so this is an interesting question, because typically when we find out where they're going and then we characterize those habitats, the characterization usually relates to the foraging prey species. So we're not really, the habitat that we're modeling isn't so much foraging habitat from the bird, it's more the habitat that the prey live in, right? So in that way, yes, we are kind of inferring differences among these individuals in areas that they target. But you know, it's this kind of trade-off between what the bird knows and expectations when it goes out into the ocean and tries to find some food, right? So it may go in a particular direction because it's had some success in that direction. So whether or not that implies that they're actually feeding on different things, not necessarily. But we are doing other things too, like there's emergence of eDNA techniques, you know, trying to understand what prey composition is in the diets of these different individuals. So this is all ongoing work, but we don't have diet, and it's a little harder to determine diet in ancient murrelets because they're not bill loading birds. So they don't bring prey back to provision their chicks, which usually informs diet in other seabirds that we study. So we have to use different techniques. And yeah, we don't have a good idea how much variation there is among individuals yet, but we'd like to definitely follow that up with maybe emerging techniques like eDNA.
Okay, yeah, I think with that, we will come to the end of today's webinar. Please join me in thanking our speakers today, Dr. Laura Cowen and Dr. Patrick O'Hara. Both are very busy people, and I really appreciate the time taken to this online webinar. I'm sure everyone enjoy the work you have done together. And
Oh,
also, I would like to thank actually our sponsors again, NISS and NISS-CANSSI, National Institute of Statistical Sciences and Canadian Statistical Sciences Institute, for coming together to sponsor this very interesting webinar series. I would also like to draw your attention, actually, our next online webinar will be on October 9th. It will be also another interesting topic, where physics on quantum machine learning. So keep an eye, the announcement will be coming soon in coming weeks. With that, I would like to thank all the participants for taking your time to attend this webinar, and have a great day.
thanks for the opportunity, Saman. That's we appreciate that.
You're welcome.
Yeah, thanks so much.